So the next speaker to honor us with her gracious presence is Dr. Ritushree Kukreti, Chief Scientist, Genomics and Molecular Medicine Unit, CSIR, Institute of Genomics and Integrative Biology, New Delhi. Dr. Ritushree Kukreti obtained her Ph.D. in 1994 from the Department of Biosciences and Biotechnology, University of Roorkee, presently IIT Roorkee. Later, she joined Laboratoire d'Oncologie Moleculaire, University of Paris, France as postdoctoral fellow and worked on understanding the mechanism of action of P53 and BCL2 in the development of human breast cancer and in vitro studies on the interactions of the reactive oxygen species with airway epithelium. In 1998, she joined the Functional Genomics Unit at Center for Biochemical Technology, CSIR, presently CSIR IGIB, Delhi, as a research associate, and subsequently in October 2002, she joined Genomed, a knowledge collaboration between IGIB and Nicholas Piramal India Limited, as a senior scientist and work on to create a powerful platform for pharmacogenomics-driven drug discovery and clinical medicine. Currently, she is Chief Principal Scientist at CSIR IGIB in the area of pharmacogenomics. Dr. Ritushree Kukreti is the author of over 105 scholarly publications in the areas of population, medical genetics, genomics and pharmacogenetics, functional genomics, and working in several related research projects. Her work significantly contributes to pharmacogenomics research in India for its implementation as clinical pharmacogenomics tests for efficient diagnosis and clinical recommendations, especially for patients suffering from brain disorders. Her principal areas of interest include human genetic diversity, the genetics of disease, and pharmacogenetics and pharmacogenomics. It's an honor to have you, ma'am, among us. So we invite you, ma'am, for your talk on cancer genomics and pharmacogenomics, moving towards personalized cancer medicine. Welcome, ma'am.
Thank you, ma'am. Thank you for inviting me for this wonderful event and asking me to say something about the experience I had in so many years. So basically, I will not be presenting the hardcore experimental data as such, but I'll try to tell you things which I have come across like a journey, what will be really helpful for the faculties how to go about in this field, because I feel that cancer or any of the disease which are very commonly happening, but they are very heterogeneous. So ideally, going like a single gene disorders, like probably we can say thalassemia, sickle cell anemia, they are having a very straightforward genomic abbreviations, genomic variations, which are targeting towards the functional understanding. And therefore, we are able to understand what genes are doing and how the phenotypes are changing. So I will probably tell you a very interesting things so that it will be really helpful for people in future to go about and take correct steps. So may I share the slides, ma'am?
Yes, ma'am, please.
Yeah. So is it clear, ma'am?
Yes, ma'am, it's visible.
Okay. So I'll start with the title, ma'am. I hope I'm acknowledging all the organizers actually to get me this opportunity. And
Thank
Yeah, sure, ma'am. So I'll be talking about the cancer genomics and pharmacogenomics, and what about moving towards the personalized cancer medicine. Of course, it's very thinking way. It is very simple. Genomics and pharmacogenomics, and like we all say, personalized medicines, but it is really very heterogeneous. And I have been working in with the brain disorders, wherein mainly epilepsies, schizophrenia, bipolar, as well as depression studies, and also Alzheimer's. But uh, as far as I understand, that genomics, when we talk about genomics, the genes and the DNA part, it is very very heterogeneous. So I'll take you first through some of the basic things, the understanding of pharmacogenomics, which we see here. There are patient pool, let's say. So we don't know. We try to give the drugs to patients, and what we see that some patients they respond towards the drug, and some patients they don't respond, and some have side effects. Now the treatment and the conventional drugs. Now, these drugs, these are not being have come through very very precise or systematic route. They are by serendipities, and by different ways we try to repurpose them also, and their dose they're not fixed, but patients they respond, and some patients they don't respond, and we need to give them the alternate drug or the dose. So what we see here, the drug specific response prediction is generally based on DNA sequence variations. What is the very easy way to go about in the field? Although we may not get the whole of the perspective of the drug response through DNA sequence, but we try to do that. And that is what we understand with pharmacogenomics. Now, when we say that this is affordable healthcare, when we say that patients they do not respond, and we try to concept that okay, fine, these are the mutations. If patients have these mutations, patients will not respond, and so you can see here we have a red core here which says that people are not responding even, and we see there is a mutation here, and people who responds there is a wild type here. So here what we say that there is a allele or genotype based prediction. Okay, so now what we see among the non responders here, we see that if we are able to change the drug dose or drug therapy, we try to understand that some of the non-responders, they change to response. So therefore, it is a wonderful way to understand that, okay, by using these mutations, genes variations, we are able to were able to have the pharmacogenomics based drug dosing algorithm. Now, if we say that pharmacogenetics or pharmacogenomics, so in simple ways, which seems to us, which we started earlier, like 15 years back, the influence of genetic variations on drug response limited to only few genes. Like, for example, we say that, okay, there are mutations in beta globin gene or some of the mutations in ADBR gene, wherein the drugs given for salbutamol for asthmatic patients, we do find some of the answers that people respond or not respond. So they are directly influence of few of the target genes. But when we talk about pharmacogenomics, what we see here that influence of many genes or entire genome, their interaction with environmental factors on drug response. But now this is very, very heterogeneous and very, very difficult to understand that how we will be able to do this for common complex disorders. So just diagrammatic picture here, what we see that there is a pharmacokinetics where the drug metabolism is involved, and there are pharmacodynamics where drug target interaction is there. Now, base of this program, we published lot of work, but now what we see here that in globally people have worked, scientists, researchers, everybody have worked globally from trying to do the research. What is the true clinical translation here? So if we see about, there are more than the 7 lakhs research papers or maybe more, 10 lakhs or more. And what is the clinical translation here? So there is a gap in translation research, and how you understand that. So basically, if we take up few genes, try to understand genetic variations, and we hope that we will get into the people who are responding or non-responding towards it is a difficult task here. So how we thought earlier about this and try to understand that why we are not able to replicate the data and try to get the more of functional understanding about the concept of genes wherein they are involved in the people who are responding or not responding. So if we to initiate that, we looked upon the radial cluster. This is the data which we did. This shows the radial cluster showing global data of pharmacogenomic research. And what we observed here, we observed that these pharmacogenomic relationships are obtained from identified 1753 genes, 53 types, 66 drugs, 4132 genes, and there are so many polymorphisms from the total publication showing biological entities and the relationship with implicated in drug response here. So, based on this enormous data, when we see what we observe, so there are different categories here. We can see here there is oncology, infectious diseases, psychiatry, neurology, cardiology, there is pulmonary, there is rheumatology, endocrinology. So there are enormous field where people have worked for so many drugs here. So what we see that oncology is being studied the most, and wherein the genes here, what people have observed are the maximum are of TPMT, CYP2D6, G6PD, CYP2C19, ESR, ERBB2, and there are many more. So these genes, they come from the metabolism genes, which we expect phase 1, phase 2, phase 3. So a lot of drug metabolism genes, they are captured here, and also the targets and transporters as well. So looking here, so what we observe that oncology is being studied the most. So here what we see, because we understand that FDA is important for us to take up these markers, and among the 2304 pharmacogenomics markers, there are 336 markers are identified for FDA approved drugs, showing which can be which can be easily going in clinical decision making and value addition in healthcare. So what we see here, so again if we observed here that cancer, which we observed there are most of the uh, mutations or the genes, the variations, they are very, very commonly and replicated and coming out to be having the clinical significance in case of people who are suffering from the disease, the types, and also the drug response. The commonly genes, TPMT, G2, which is transporter, EGFR, growth hormones, growth receptors, then phase 2 enzymes, and more. What we see here among 336 markers, still, I'm talking about the gap in the translation research, wherein among these 2304, only 33 markers are being applied in healthcare for among all the disease aspects. So only 15% is actually is taking up to translation as of now globally. So taking forward in showing with the oncology of related cancers, I would like to show some of the important data, which you can say are lower hanging fruits, wherein there is the disease is so heterogeneous, but we try to understand the basic part of cancer, that how if we are able to dissect the heterogeneous part and try to understand the diagnosis of the cancer type, whether we are able to understand the drug part or what type of treatment we should give for patients individually here. So I'll take up this particular data, which is coming out from our SNP project, which we did in 5 years before. It was a consortium prepared by Professor Brahmachari and which was executed by Mithali as the convener, and all the members across CSR labs were the participants for these, and we have taken up a lot of understanding of genetic variations here. So if we talk about here, so I'll take up an example of the cancer type where the thyroprine therapy is being given, and we looked upon the TPMT alleles, which is again thyroprine methyl transferase, and this is very, very important actually in global understanding, where the TPMT gene, the allele present here, is involved in variability in metabolism. So we checked this data across Indian population, and what we observed that there are genotype, which is wild type across wild type mutant and mutant mutant. So this shows the functional understanding that there is extensive metabolizer, intermediate metabolizer, and poor metabolizer. Now here, based on this, there people are poor responders, good responders, and of course there are drug toxicity. So I'll take this thing towards the understanding of predictive value of risk of ototoxicity in pediatric patients. So we can see here a map which shows that their wild type, which is across the Indian population. We can see here wild type and mutant, which is black, is some of the places, and the mutant, which is red. So what we have understanding that we can of course establish diagnostic values looking at thyroprine response markers and showing measures of clinical utility here. How that there is a drug which is given and there is a ototoxicity involved here, and the people of this particular population are at risk. So we have looked upon the combination all the combining all the data globally and found that there is a high specificity and sensitivity positive predictive value for this particular genetic variations with functional understanding. And this is what a very important data across Indian population as well as global population, and which shows the risk of TPMT 3 star 3A risk genotype in Indian population. So if we talk about now taking up this understanding that, okay, we will take up genetic variations at the understanding of functional level of the gene as well as the phenotype contribution. So taking up this, I'll just go through that what the understanding of cancer is and how it can be taken up in terms of genomics and pharmacogenomics. How diagnosis of individual cancer, which is so heterogeneous, can be taken up, and if genomics can help us further to understand the treatment, and therefore we are able to have a idea of responding towards the drugs or whatever the therapy is being provided. So there are three major stages of cancer hallmarks: the reprogramming cell growth, which is expected from the genetic changes which lead to immortality, birth and survival of the formed cancerous cell, which is the first one, and the proliferation, invasion, and metastasis, which is the important part. And thus, once the malignant cells is formed and has survived the first round, the invasion, the metastasis may be followed as an inevitable consequences. So if we talk about, there are lots of more than 500 cancer types based on the cells they arise. And there are carcinomas, which is 90%. As such, blood cancers, which is 8%, and sarcomas are rare, although. So head and neck, lung, prostate, breast, esophagus, colorectal, anus, etc. These are the major carcinomas happening, and the blood cancers, leukemia, lymphoma, melanoma, and muscles and nerves, which are very rare sarcomas. So if we talk about the common cancers here, so the cancer, which is the cause of the death worldwide, accounting for nearly 10 million deaths in 2020. The most common in 2020 are the breast, lung, colon, rectum, prostate, skin, and stomach. In India, most common cancers for males are lung, prostate, tongue, stomach, constitute 36% of all cancers. For female, the breast cancer, uteri, ovary, and lungs constitutes 53% of all cancers. 1 in 68 males in lung cancer, 1 in 29 females, and 1 in 9 Indians will develop cancer during their lifetime. And which is again 0 to 74 years of age. The projected incidence of patients with cancer in India for the year 2020 is 94.1 in 1 lakh for males, and females is 103.6, the incidence, which is the new cases yearly. So, if we talk about the cancer genomics, which tells us that the genome is responsible, genome of the cells in the body, leading them to grow uncontrollably. These changes involve DNA mutations in the genome, and the expression differences between tumor cells and normal host cells, which are very important in cancer genomics. So it is aimed to understand genetic basis of tumor cell proliferation and the evolution of cancer genome under mutation and selection by the body and the immune system and therapeutic interventions. So cancer genomics actually focus on finding new cancer genes, that is cancer drivers, identify molecular signatures to stratify tumors. So now understanding these findings that cancer is so many types, so we need to understand the individual. It is not population based cancers also, the tumors. So we need to understand individual type of cancer or tumor. To understand that, we need to understand at molecular level. So what are the genetic structures to stratify tumors actually. So now here it will be really helpful for us to understand that if molecular signatures, the molecular strategy genomes, they are having some clue at the level of serum or blood, so that we can understand how it can be developed to biomarkers to stratify tumors also. So this is the first understanding which we need to have in terms of cancer diagnosis. And then finding new, when we understand the type of cancers, the cancer diagnosis, finding new therapeutic targets are important. So therefore, and what we move here to personalized cancer treatment. For example, like if we are taking up the cancers, and if we take that inflammatory markers are high in that case, so whether we are able to have those inflammatory markers as the biomarker to understand that how the pathways are influencing and how they are coming out as a biomarker in blood or in serum. So that is one important fact which we should understand and we should go about. So I'll talk about major events in a decade of cancer genomics, whereas DB-SNP database, which is very, very important. However, there is enormous billions of variations in genome, but they are non-polymorphic, and only polymorphic variations we need to tap, and we need to understand their functional effects. So these are the milestones here. And then after DB-SNP's database establishment, Human Genome, HapMap project, and Code project, which tells us a lot of understanding about regulatory. Then the Cosmic Initiative programs, where we observed that lot of somatic mutation, apart from germline mutation, there are lots of somatic mutations happening in cancer. And then there is a Atlas program of cancer genomics being established here in 2005. Then comes lots of mutations in colorectal cancers, then whole exome sequencing being established. And therefore, like this, we can see the next generation sequencing have come up, and there are lot many things. But based on this, based on these events happening, a lot of data being done, apart from these facts of projects and consortia, project and major events, the timeline of most remarkable precision oncology highlights also, which tells us about landmark therapeutic molecular targets. Here we can see here that there is a ER for tamoxifen, HER2 for trastuzumab, then BCR ABL, then EGFR for another drug. So these are the molecular targets for common anti-cancer drugs identified, then different diagnostic technologies also, and regulatory aspect, regulatory landmarks, which is government regulatory policies that aim for cancer therapeutic applications. So based on this, from where this is coming up as a highlights and timelines, there in due course of time the cancer projects and consortia are being formed, like the patients cohorts has been developed. And what people have what here the consortias, the scientists and researchers, they have expression patterns, copy number variations, somatic mutations, germline variants, epigenomic profiles, and structure. So these are being developed and taken up as a consortia format and being in totality being done. So what are here? The Atlas program is telling us that we have tapped upon the mutations, copy number, gene expression, DNA methylation, microRNA, everything they have kept here for samples, gene loci, and the individual type of mutations. So what we talk about here, we have thematic pathways here. Now these thematic pathways, what we tried, as I said earlier, that we tried to find the most of the data, but then how to streamline that data, the heterogeneous data, of course, and how we come out with the biomarkers out of these. So from all this data of consortia and projects being collected, so what we see, the mutations associated. which comes on oncogenes, tumor suppressor genes, DNA repair genes, then carcinoma activating genes, deactivating genes, cell cycle checkpoint genes. So these are the list of the genes which carries mutations based on the study so far. So if we look about genetic testing for breast cancer, so here this slides probably will tell us that there are high risk of breast cancer genes, which are of course BRCA1, BRCA2, P53, P10, and PALB2. So here, this is a diagrammatic picture in circle. We can see that this high risk of breast cancer genes will depict from the red color. So there are moderate risk, which is the again the blue color depicting the uh, moderate risk again for breast cancer. So these things, these genes coming out not only from one study, but is the global projects and consortia data which is being evaluated for different types is being collected. So now these mutations which are coming out to be high risk, moderate risk, or low risk, these are being used as panel for now. So there are lots of cancers. I just showing the example of breast cancer, but there are lots of cancers which, as of now, not only one mutations, there are more than 100 of variation panels are being used to depict the risk of cancers, individual cancer type. So what we see here, getting to the point where we could actually use the genetic information to make the prescribing decision requires extremely high level of evidence, replications, validations, and mechanistic studies. So what we see here, looking at this particular uh, data, so, what is the need at present in India that, of course, such kind of data we do have from the other parts of the world? So, we need to understand the what is the mutations or panels we can develop for our individual cancer types. So based on the data which the global data shows, which is being depicted, the summary of that is basically there are the cancer causing mutations. If we talk about, there are very limited high risk cancer mutations are being shown for high predictability, high replicability, and with the correct treatment. So the cancer causing mutations for drug treatment available is HR plus, HR2, HER2, PIK3CA mutations, and BRCA mutations. For this particular mutations, there are personalized care for the drugs, the Piqray and Lynparza. And the mutations with no drug available, the people, they try to give the radiations or chemotherapy. So as such, if we talk about that this particular high risk mutations are being targeted with the drug which is personalized. So this data, just to show here that here, if you see there is azathioprine, irinotecan, and 5-fluorouracil. These are the three important drugs. Just to show that FDA has approved the TPMT STAR2, STAR3A, and 3C genotypes for particular toxicity. And also the phase 2 enzyme UGT1A1 is also used for the allergic reactions or for side effects, as well as 5-fluorouracil is being used again for the marker here, TPD deficiency marker here being used for the side effects, which is myelosuppressor, suppression, mucositis, dermatitis, diarrhea, and cardiac toxicity. So these are the well-known facts for the drugs given for the particular cancer. So what approach we talk about now, approach for disease type understanding and drug response marker development, there is a clinical understanding. Demographic factors, environmental factors, disease phenotypes, serum drug levels, transcriptomic, gene expression profile, DNA sequence variation, genetic is exome, whole genome profile, SNPs, copy number mutations, variations, mutations, and drug response here, genomics and pharmacogenomics signatures. So this may illustrate the effect of genes, genetic polymorphism upon the DNA variations in disease drug response to improve types and pharmacovigilance capacities of drug responses. So what we understand here, it is thinking is this, that how the clinical important factors can be taken up to define the disease type, of course, at DNA level also, and then try to understand the gene expression, then serum biology, introducing the biomarker as well as the genetic, including the genetic aspects. So based on this, what we have worked upon is number of brain diseases, especially the epilepsy program, and what we have resource development, that here that there are more than 4,000 detailed phenotype patients what we have enrolled so far for 15 years follow-up with repeated refinements to develop endophenotype and healthy volunteers which have participated in different studies. So we have created a catalog of prioritized genetic variations and also with their functional aspect, which are giving us association with phenotypic definition across different cohorts. We have generated genomic data, which is included in global meta data from other people also. They have included our data of drug response studies and cited our work also. We have established the collaboration multicentric prospective studies across Indian collaboration, also few of the international, trying to set up the international collaboration also to answer the criticism of allele frequency differences that occur due to ethnic origins and establish these diagnostic markers in ethnically diverse Indian populations so that we can replicate the data. We have also done functional assay studies to assess the impact as biomarker, and we have developed the chip for particular drugs for the epilepsies and also for the depression patient. So we have actually published in epilepsy pharmacogenomics, schizophrenia, Alzheimer's, depression, Parkinson's, dementia, asthma, and others in pharmacogenomics. So I would like to actually say, acknowledge basically all the collaborators which actually helped us in our studies, which we have actually tried to resolve the heterogeneity among the common complex diseases which are across Indian perspectives and also the international. And I would like to actually acknowledge the students, the lab students, the lab faculties, and of course the funding agencies which have actually helped us for giving us funds here, like ICMR, which has been very supportive for us to give us the pharmacogenomics projects, and then DBT, DST, and of course CSR. Thank you.
Thank you very much, ma'am, for enlightening us on cancer genomics and pharmacogenomics, cancer-associated mutations, and targeted cancer therapy. Your kind presence in the program has encouraged us a lot, and we are looking for now questions. So we have one question from Mr. Abhijit Devnath in the chat box. So the question is, For personalized medicine, which one will you prefer? Small molecules based drugs or large biomolecules like antibodies?
See, that is what I have been looking at in my studies, that it is something which is a type based. So the problem is, like, we cannot say as such that small molecules or antibodies as such, the antibodies, as you said, is maybe helpful for one type or the other. So we need to check the individual type for this, I believe, to go forward and have it. Because what we see here, that there are drugs, even small molecules, there are drugs which are not being actually giving response because of patient's heterogeneity even.
I hope Abhijit sir is satisfied with his answer. Yes, he's nodding. Our next question is from Mr. Surjit Pathak. Mutation-based biomarkers in precision medicine in cancer diagnostics. I hope the question is incomplete.
If I
Yes.
want to take up this, so
Hmm.
therefore I have actually mentioned here that what we have observed, for example, I will talk about TPMT as well as the breast cancer also. What we see here, whatever the markers in is being replicated in multiple populations are very, very important, and we can be taken up for the understanding of risk of cancer as well as diagnosis of purpose and for personalized medicine as well. So I have looked upon that there is a panel for breast cancer, colorectal, and many more cancers which are very much available, which is based on the studies happening. Studies, it is not very much true that it may be present in your population also. It is not that. So those markers which are very, very important can be taken up for personalized medicine, but we are not sure that that will happen to your, because see, when we talk about mutations, the allele differences, they are happening in our population. So allele frequency, which is happening in our population, is telling us that there are possibilities that markers are lying there. So we need to again go the whole exome sequencing and next generation sequencing to tap more of variation in those genes as well, more, so that we can come out with the better personalized medicine.
Thank you, ma'am, for the answer. So our next question is from Dr. Soumya Das. Is pharmacogenetics a personalized medicine?
Pharmacogenetics is the personalized medicine. Okay, so I will say like this. That was my third slide, I guess. I can again showcase that when we talk about personalized medicine, it is something individual, right? Individual. And when we talk about population based medicine, it is something population. So, as of now, what I feel, pharmacogenetics can be population based medicine. Population based medicines is such that the variations what we see in the population, they are present in groups. So let's say I'll take one example here, that if the mutation or a polymorphism is there in the population among 20 people out of 100. Now those 20 people out of 100, whether they will, if they are not responding to particular drug, if they're not responding to particular drug, it is really a chance, it will known as mutation otherwise, that there is only 1% in 100 which is having that mutation, and what we tap here is that is associated with poor response. So what we are tapping here is the general genome analysis and the general frequency of the mutations, or I should say polymorphisms, so that we can find the people in population, they're responding or non-responding. I hope I'm able to make you understand. In cancer, it is much more heterogeneous. So pharmacogenomics, it is much more heterogeneous. Pharmacogenomics is something which is population based as of now.
मेंशन नहीं कर
Population.
दिया, चेक कर लो एक बार.
वो आपने शेयर करा है ना ग्रुप में. Thank you, ma'am, for the answers. I hope there are no more questions, and if there are some, then we'll get back to you.
Sure,
Certainly.
ma'am. I will be very happy to answer, because it is something very, very important to convey, that it seems to be so simple, but I want to, it's my last conveying sentence, that drug metabolism, it is so flexible that what we eat, what the environment is actually determines everything. So no matter what, your genetics, what percentage it is playing a role. When your gene is inducible, your gene is reduced activity based on your environment, based on what you eat. Even if you take the valproic, I must say, I'm just taking one of the drug, or any cancer drug. If you take that drug, you actually, your body induces the metabolism to take it out, to excrete it out. So what genetic variations will do here? So it is something very, very tricky, and understanding the subject of genomics and pharmacogenomics, so that we kind to have a clear picture about the genome and pharmacogenome, I should say.
We have one more question from Dr. Surjit. It is related to KRAS mutation in colon cancer.
Yes,
The
yes.
question is, yes, ma'am.
I believe this is actually involved in the panel. This is very, very important, and it is being used in the panel for the cancer, you said. So you want to have some specific important thing about it? Can you expand it?
Ma'am, the question is not elaborate. I think we can get
No,
back to it
no.
some
Sure,
other
sure.
time.
Yeah, yeah, yeah.
Thank you, ma'am. Thank you so much. Ma'am, Rupa ma'am wants to say something, but your speaker is not on, ma'am. It's muted. Yes, now you can.
Thank you so much, ma'am, for a very wonderful presentation.
Thank you, ma'am. Thank you so much. It is
Yes.
so much of, I mean, kind of you to ask this, because it actually not working in as such in cancer, but I
Yes.
must say the other disease aspect, cancer is far more heterogeneous
Of
to
course, ma'am.
have that understanding. So it is very, very important to take a correct step to go about in the research projects, so that we just not just keep on rotating the wheel every time. So I must say that we should actually share our thoughts and our understanding to our juniors, as you are having such a wonderful program of faculty development. So they should actually be aware of what actually people don't discuss about their journey as such, what is the negative points they are coming across. So that is also very important, I guess, for new beginners to go about it.
Thank you so much, ma'am. Hope we will get you in our future programs also with us.
Yeah,
Thank
sure, ma'am. So nice. Thank you so much, ma'am. Thank you.
Thank you, ma'am. Thank you so much.